Hello, hello. Welcome back to another exciting part of our audio lectures. We have made it to our last part of Unit 2, and I think we're moving along quite nicely. The only thing left to do is to take a look at Chapter 12, which I think is a very interesting one because it's going to take a look at the overall shape of chromosomes, and we're going to discuss how do you go from a single strand of DNA to a coiled up version of your chromatin and keep condensing it down until you get that stereotypical chromosome structure that we're so used to seeing when we take a look at a karyotype map. Okay, so chapter 12 is all about the chromosomes I was mentioning to you before, and I feel like by now we're becoming experts into what a chromosome is. We know that it is extremely condensed DNA. We also know that it has proteins that it packs in there. So for instance, you might recall your histone proteins that you coiled your naked DNA around when you were making your nucleosomes. And we also know that the chromosomes collectively will make up our genome, which is our genotype, all our genetic material that we need to utilize. So what we'll do in chapter 12 is we'll start taking a look at how bacteria set up their chromosomes and what exactly you will find in there and where they're housed and all that other good stuff. And then we'll wander over to the eukaryotic cells. And since eukaryotic cells not only have more DNA than the bacteria, we also have to take a look at alternative places that they store their DNA. In addition to the nuclear chromosomes in the nucleus, we also talked about things such as having extranuclear inheritance of DNA, such as the one that you find in the mitochondria, as well as in plants, the one that you will find in chloroplast. Now, bacterial chromosomes are relatively very simplistic in their structure. Bacteria usually have a single chromosome that will be in a circular format, and usually what we see happening is that they won't be all that long, but it is very common for them to have multiple copies of that one chromosome floating around within their cytoplasmic region. Now, when you look at the chromosome of the bacteria, as I mentioned to you before, oftentimes it will be in a circular format. And when we look at the gene map of the chromosome, you're going to notice that there tend to be different sections of it labeled. One of the sections that you're going to see labeled are what we call protein encoded genes. Those are going to be the sequences of DNA that are able to do transcription and translation. Hence, they're able to go ahead and formulate proteins for the cell. You will also have what we like to call introns or intergenic regions. Intergenic regions, as the name indicates, are going to be in between the genes. These are areas that have DNA in them, but the DNA cannot be utilized for transcription. Some people used to call this junk DNA. However, upon further research, now we know that it's not really junk DNA because it does play a role in the overall folding ability of the DNA. It helps with DNA replication. It is evidence of evolution as to the replication cycle, and it will also contribute to some of the genetic recombinations that you'll see as you're creating variety from one generation to the next. So people used to call it junk DNA because it didn't do transcription, but we now often know that it will contribute in other ways to the well-being of the organism. Now here's a very simplistic illustration of that circular chromosome that you'll come across in a bacterial model. And what we can do is we can go ahead and look on the bottom left-hand side where we're going to see a little key so that you can figure out what exactly all these lines mean. Now, all the solid lines are going to indicate the gene regions. So those are the regions that are able to do your transcription and encode your proteins. And then you're going to have those intergenic regions that I was telling you about. They're indicated by the slight red pinkish segments. And then you also have repetitive sequences. Now, repetitive sequences are usually just dispersed throughout the chromosomes. Some people believe that they're there as evidence of evolution. So every single time the cell replicates, it adds a little bit to its DNA length, creating repetitive sequences. Some people also believe that they'll help with the actual formation of DNA. So it's essential for balancing out when the cell goes, in the case of the bacteria, through binary fission. So on the side right here, on the left-hand side, you're also going to see an origin of replication. And in fact, regardless if you're looking at a bacterial chromosome or a eukaryotic chromosome, you're always going to have at least one origin of replication, because that will be the site where the DNA replication will be initiated when it's time for the cell to start its overall division. Notice also that if you come over to your right-hand side, you have some of the key features. And it reminds you that most of the bacterial species will have a circular DNA. Usually it's not all that long, that they have a single type of chromosome, but it's quite common for them to have it in multiple copies. And as you can see from the key dispersal around the chromosome, they remind you that it's a combination of genes as well as intergenic regions, which are the short sections with the non-transcribable genetic sections. Also, you need an origin of replication, because that is going to initiate your DNA replication. And then you have your repetitive sequence that will also be dispersed throughout the chromosome. This is a really nice picture. It kind of shows you how the DNA in the bacterial cell is being clustered together. Now, please remind yourself that bacterial cells are examples of prokaryotic cells, so they do not have a nucleus. So instead, what they'll do is they're going to house their DNA in a nucleoid region. A nucleoid region is basically going to be a section that is not very organized, but it will keep the the chromosome together. One thing that's very interesting though is that the nucleoid is not really an organelle, which means that it doesn't have a membrane, which means that the DNA comes into actual physical contact with the cytoplasm, which is obviously the inside material of the bacterial cell. In the picture, I do want to point out that you can have several nucleoid regions. Each of them can have one or multiple copies of that one of the same bacterial chromosome. Now, we do see that the bacteria cell has a massive amount of DNA for its size. And what we do need to see is very similar to when we were talking about our chromosomes, is that we utilize the concept of creating a chromosome from a single strand of DNA, all in the effort to compact and be able to house all this massive amounts of nucleotides. So on this particular slide right here, it tells you that on average, if you want to fit the chromosome DNA that a bacteria has into its prokaryotic cell, we're going to have to compact it about 1,000-fold. That's a massive amount of compression that you're going to have to do. So how exactly are we going to do that? Well, the bacteria will utilize different DNA binding proteins in order to compress and coil up the DNA. So what you can see from our illustration at the bottom right here is that we have our nice circular chromosome DNA that we're used to seeing when we were chit-chatting about our bacteria. And what we're going to do is, in order to compress it, we're going to utilize these DNA binding proteins that will start looping together the chromosome by basically anchoring the bases to the loops that are in the center of the drawing, in the center of the plane right over here. And as it's creating these loop domains, we're able to compress it down. And most of the time, it's about a 10-fold compaction that you see. So it's a nice amount that it starts to dwindle down so that it can accurately fit. But it's not enough. So what we see happening is that we're going to need to do additional coils. As these additional coils start to form, we're then going to go ahead and call them supercoils. DNA supercoils are going to generate lots of twist because of the force that's being expanded on them. And as these additional coils are given in the twisted formation and you start supercoiling your DNA, you further compact it down so that it can properly fit in the aliquoted nucleoid region. So look at it go from the loop to the supercoil. And as you can see, you're just making extra coils inside of the loop, hence the name. Now, also what we see happening is that you can have what we call negative supercoils. And negative supercoils basically indicate that you altered the direction of the supercoil. So what do I mean with this? Well, most of the time as you are coiling, we tend to coil for the negative supercoil, we tend to coil towards the left. And as we are you know what, I'm sorry, let me rewind that for a second. When you start coiling up your DNA to make it a supercoil, usually what you do is you rotate to the right. And as you're rotating to the right, you're creating those supercoils that we saw on our previous slide. However, if you create a negative supercoil, you coil to the left. And as you're coiling to the left, what's going to end up happening is you're going to create a section of negative supercoil, which is what you see happening over here. So a section that kind of loops out, that's a little bit out of place. But it turns out that you need this negative supercoil, this coiling with a left-handed twist, because it's going to not only help you further compact your chromosome, it will also create enough tension that when it's time for the DNA to replicate itself, it is very easy for the double helix to completely separate itself, so that we can do our DNA replication, and also that the genes are always available for transcription and for translation. So I'm sorry that I started off this slide a little confusing, but what I was trying to tell you is that when you do your supercoil, you normally go and you do a right-hand turn, and this just keeps overwinding it. But what we see happening is that to make sure that as we're coiling it, we can still utilize the chromosome for its function, which means access to the genes, we're going to also have to go ahead and do some left-handed turns, and these are going to create negative supercoils. So left makes the negative supercoils, and this allows you to create an area that can still be easily accessed whenever you need to do DNA replication and transcription, because the double helix will be able to separate into its individual strands. In order to assist with the supercoiling, what we see is that there are a handful of enzymes out there that will be able to regulate the negative and the positive supercoils that the bacterial chromosome will utilize. Two of the most common enzymes that you're going to come across are going to be called your topoisomerase 1 and topoisomerase 2 enzyme. Topoisomerase 2 enzyme is also called the DNA gyrase. And what this particular enzyme does is that it's all about generating negative supercoils. So it can generate your negative supercoil. It can go ahead and keep intertwining your DNA, but we also know that it can go ahead and change its overall rotation, thereby allowing some of the relaxation of the positive supercoils. So it can generate the negative supercoils by rotating more towards the left-hand side, but it can also stimulate relaxation of the positive supercoils, which are the more the right-handed rotation. Topoisomerase 2 is also going to assist with the supercoils. It, however, is more about relaxing the negative supercoils. So topoisomerase 2 wants to relax the positive, topoisomerase 1 wants to relax the negative supercoils. And the way it's going to do that is that it actually will physically break the DNA molecule. It will rotate it into the position that it requires, and then it will recruit over a ligase enzyme to seal it back up. So that's very unique about the DNA topoisomerase 1, is that it will physically pull apart and break one strand of DNA to get it to properly orient itself to the coil that you are looking for. Now, a eukaryotic chromosome is going to follow slightly different rules. Part of that is because eukaryotic cells will have multiple copies of chromosomes available in their nuclear region, but also because the chromosomes from eukaryotes tend to be in a linear format. Part of this will be seen in the fact that we have a lot more DNA in a eukaryotic cell than in a prokaryotic cell. And also what we see happening here is that within the DNA segments of the chromosome itself, we're going to have to account not only for the origin of replication, very similar to what we saw when we were looking at our circular chromosomes for our bacteria, but we also have to discuss the function of the centromeres and the telomeres, both terms that hopefully you'll recall from our previous unit. All right, now the origin of replication will, very similar to the bacterial chromosome, will be the site where DNA replication will be initiated. One difference that we see here, though, is that prokaryotic cells, or I should say bacterial cells, tend to only have one origin of replication in their circular chromosome, whereas in eukaryotic chromosomes, it's actually quite common to have several of them. You will also remember that your chromosomes have a centromere. This is going to be the section where we house together or where we physically connect our sister chromatids. So it is essential for aligning the chromosomes and in essence when they have to line up down the equator in metaphase and start physically separating their sister chromatids in anaphase. To assist with this movement during mitosis and meiosis, we see that there are kinetochore proteins that are going to be sitting very closely to the centromere, and these kinetochore proteins will be guiding the spindle process. The spindle is the protein mechanism that will physically be moving around the chromosomes as we align them and then separate them into their separate batches in both mitosis and meiosis. And then last but not least on the list right here, we have our telomeres. The telomeres, I want you to think about them as little caps. They sit at the end of the chromosomes. They are excellent in maintaining and preserving the length of the chromosome. And it's also one of the ways that your body tries to prevent translocations from occurring. Translocations are when we talked about chromosomes abnormality, when you get quote-unquote jumping genes, when a piece of the chromosome will break up and randomly wander to another non-homologous chromosomes. So here is the illustrated example that goes with all those different key terms. So let's see what we're looking at. So we have our different origins of replication. There you go. So you have 4 of them pictured in over here. We have our centromere in yellow that we're going to utilize for the segregation of the sister chromatids during our mitosis and meiosis of our cell cycle. We have our kinetochore proteins, which are going to guide the spindle also during mitosis and meiosis. And then we have our telomere sections at the end cap. Let's look at some of the key factors. It says that the eukaryotic chromosomes are usually linear. Gotcha. They tend to be longer than the bacterial. They tend to occur in sets. Part of that is why we utilize, excuse me, things like karyotypes so we can follow the maternal and paternal alignment of these homologous chromosomes. We also see that very similar to the bacteria that you have areas that have been marked as genes. So that's the parts where you're going to go ahead and do your transcription and translation. We also see that you have areas that are labeled as repetitive sequences. Notice how they are heavily populated by the centromere and the telomere sections. You have lots of repetitive sequence there. And you'll also have some dispersed throughout the length itself. And what else do we have going on? Oh, we have our telomeres. We mentioned that. Those are the end caps that are protecting the length of the overall chromosome. So a little interesting fact about telomeres is that it turns out that every single time you replicate your cell, your telomere gets a little bit shorter. And at a certain point, the telomere is so short that the DNA can't replicate it anymore. Unfortunately, this is one of the reasons why we start to age and eventually we'll come to the point in our life where we can't produce any more cells and will die of old age. Certain cells in our body have a telomerase enzyme, and this enzyme is able to keep the telomere at a consistent length. You would see it in cells that have an unlimited ability to divide. So for instance, stem cells have a telomerase enzyme. So they have unlimited amount of replication that they can do. You also have, for instance, the stem cells that you have in your skin, your bone marrow cells that help you with your red blood cell, white blood cell, platelet production. Guys, your sperm cells also come courtesy of a stem cell. So until your dying day, you'll be able to produce sperm. And cancer cells. Cancer cells have telomerase activity, and that's in essence what makes them so hard in essence to treat and always kill off, because they have this unlimited ability to just keep replicating, and they're not limited to the length or the cap of the telomere that's placed on the majority of our cells. All right, now here is just some of the written information of what I've mentioned to you from the illustration before. So we see right here that we have the genes that are going to be dispersed between the centromeric and the telomeric region. So that's between the centromere and the telomere, the connection point. A chromosome can have anywhere from hundreds to thousands of genes. In humans, for instance, we have anywhere between 22 to 23,000 genes that have been dispersed among our 46 chromosomes, or the 23 pairs of chromosomes. At the bottom right here, I don't think it will surprise anybody to see that in more simplistic or lower eukaryotes, usually the genes are relatively small and they'll have few areas of introns. Introns are non-coding sections of the DNA. So the introns are areas that usually are your intergenic regions. They do not code anything. And if you come across the term exons, E-X-O-N, exons are usually the areas that pertain to the gene, where you can have coding and transcription occur. So introns don't do any coding, whereas the exon sections do have the coding because they have the gene. At the bottom of the slide, it tells you that a higher eukaryote, so if we're looking at more complex organisms, then we see that the genes tend to be a little bit longer. We tend to have a little bit more introns. And part of that will come overall from the length of the gene that we are looking at. Now, believe it or not, genome size is not always an indicator if we're looking at a more complex or more intelligent organism. What you really want to look at is the ratio of these exons versus intron regions. So how many genes versus non-coding sections do you actually house within the genome? And in fact, what we see happening over here is that within our eukaryotic genome, there is a great variety of different sizes. But just because you have variety doesn't mean that it's going to be correlated to the level of complexity. And a really good example that they have is the one that's in the picture right here. You can see that we're looking at two different species of salamanders. And the one at the bottom has a genome that's almost twice the size of the one that's on top. However, from a phenotypical aspect, what we're seeing is they did look very similar and they have very similar behavioral attributes. So you wouldn't really see a significant difference in one having twice as much genome as the other. And keep in mind that all has to do with, just because you have a lot doesn't mean that it is quote unquote quality DNA. A lot of times with a larger size genome, you might just have a lot of repetitive DNA sequences that would fall into the exon sections that do not code for any protein. The only way that you would have a correlation between genome size and increased complexity, if that would indicate that you have more genes than the smaller genome, because the more genetic regions you have, the more you're able to transcribe and create different versions of proteins that need to be utilized. All right, in fact, let's take a look at this. So normally what we see happening is that within the human genome, you'll have different classes of DNA sequences. And what we see happening is we can have 4 separations. You have regions that are encoding the protein, so your little exons. Look at this number, only 2%. Then you have 24% that are a combination of just introns and other segments of DNA. And then you have unique pieces of DNA, but they're still non-coding. So collectively, altogether, right over here, we have our unique segments that make up about what, like 41%? Yeah, 41% of our overall genome. So the other 59% is repetitive DNA. So repetitive DNA meaning that it really doesn't contribute anything to our transcription and translation abilities. These can be just anything from like extra copies to just, repetitive sequences of nucleotides. Either way, we do not get a benefit from it. And this just kind of correlates to what we were talking about on our previous slide, in the fact that just because you have a more complex or a higher amount of genome doesn't necessarily mean that you get a greater benefit out of it. All right, so let's talk about compaction. How do we wind down and compress these massive amounts of DNA that we have into our 23 chromosomes? So take a look at some of these numbers. So it says, if stretched end to end, one set of human chromosomes would be over 1 meter long, whereas a nucleus is only about 2 to 4 micrometers in diameter. So obviously it's not a comparison, right? We have a massive amount of DNA to fit in a small place. And thus far in our discussions, we have talked about that DNA will go ahead and start coiling up. And one of the first things that it'll do is it will make chromatin. And the chromatin is when you're well on your way to pressing down until you get to your coiled up chromosome. But it turns out that there are a lot of additional steps that come into play when you go from naked DNA all the way over to your chromatin and your chromosome. So what I would like to do is, over the next 2 slides, I want to take a look at those steps, and I also want to take a look at those histone proteins that are going to help us with compacting our DNA. So remember how we talked about nucleosomes before? We talked about it in one of the first few chapters that we handled in Unit 1, and we talked about the fact that if you have naked DNA, what you can do is you can go ahead and you can take 8 histone proteins, and you can wrap the DNA around them, and as you're wrapping the DNA around them, what are you creating? Your nucleosome. And the nucleosomes are the ones that will stack on top of each other, and they'll start looking like that old school telephone cord. That's when you get chromatin, okay? Now, what this slide takes a look at is what those histone proteins are actually bringing to the table. So it turns out that you're going to need 8 of them, an octamer. Out of the 8, you have 4 different ones that are paired up, and those are going to be H2A, H2B, H3, and H4. The H obviously standing for your histones. And on average, if you create your octamer, so 4 of them stacked on top of each other, you can wrap about 146 base pairs of DNA around that complex, creating your nucleosome. Here's what it looks like when we zoomed in on it a little bit. So you can see your histone octamer. See how the circles are color-coded? So we have our blues, we have our greens, we have our reds, and our khaki. Notice how there are 4 different types, H2A, H2B, H3, and H4. All of them are paired up, and we now see that we have a nice strand of naked DNA that is wrapping itself around the octamer And as I said before, holding about 146 base pairs of DNA. So 146 of A's, T's, G's, and C's. All right, so it's very similar to kind of like putting a string, like using beads to kind of coil up a string. And when it does so, it shortens the DNA about 7 folds. Not a lot, but we're on our way. Okay, that's a good thing. All right, and also notice how in between the nucleosomes, you're going to have what we call a little linker region. So you're going to have a little piece of naked DNA that is accessible. All right, so let's see. It can change the length from 20 to 100, so it depends on what section you're looking at. And then here's the diameter, about 11 nanometers. All right, so we have folded it down to about a 7-fold change. The reason that they like to use histones when they create our nucleosomes, it's all because of the fact that they contain a lot of positively charged amino acids with an emphasis of lysine and arginine. And because of this, it's going to have a good time binding to the phosphates that make up the backbone, because they tend to have more of a negative charge. And these histone proteins will go ahead and have a globular domain, and most of them tend to have a little terminus or a little tail that's also charged, so it will be able to recruit over the naked DNA at a more efficient rate. As I mentioned to you before, within the octamer, you have 2 sets of H2A, H2B, H3, and H4. These are what we're going to call our core histones because they make up the octamer. And then you have a 5th histone. That one is called H1. H1 is called a linker histone because it's going to be binding to that linker DNA region. You know, that region that's naked between the 2 nucleosomes. It doesn't bind as tightly as a core histone, but it is definitely important because it will allow you to still participate in chromosome compaction, but also have easy access to the DNA, so you can still utilize it for gene expression, such as transcription and eventually DNA replication. All right, so here's our illustration with our linker protein, and I know there's a little box. Here you go. So it tells you that don't ignore the histone H1 because that is our linker protein that still plays a very important role in the compaction of the chromosomes. It just simply is not part of the nucleosome. The nucleosome is just going to be the octamer with the naked DNA wrapped around it. Alrighty, so you have taken your naked DNA, you have wrapped it around the octamer, you have made your nucleosome. So now we have compacted down to about a 7-fold, and we're going to keep going. And what we see happening is as we start stacking up the nucleosomes on top of each other, the next anatomical structure that we're going to encounter is going to be the 30 nanometer fiber. And this fiber is going to continue compacting the DNA by another 7-fold. And depending on the turns that it will make, it is either going to have a solenoid model or a zigzag model. And they're not really sure, so notice how on the PowerPoint it says it's been proposed. Because part of what happens is whenever you remove the DNA out of a dead cell, the structure will automatically alter. So it's really hard to kind of see this in a living cell versus a dead or harmed cell. But either way, let me show you in the next picture what the solenoid model and the zigzag model of the 30M fiber looks like. So here you go. So on your left-hand side, you have the solenoid. This tends to be more of a very structured setup. It's going to have a nice little spiral, about 6 nucleosomes per term, whereas your zigzag model will be a little bit more spaced out, allowing for the configuration to have not as much face-to-face contact or physical contact with the nucleosomes. Notice how regardless of which model is used, the width will still be at the 30 nanometers indicating the name of the fiber that we're looking at this compaction level. So we're not done yet. We still have a way to go. But at this point, what we need to do is that if we combine the workings of the nucleosome and our fiber, then altogether, we've come down to a 50-fold decrease in size. So we're doing well, we're doing good. But we need to keep going. But in order to compress the DNA even further, or I should say the chromatin even further, at this part, we're going to have to interact with the actual mechanisms of the eukaryotic cell. So what we need to do for further compaction is we need to go ahead and take a look at the nuclear matrix. So what's happening within the nucleus? And we need to take a look at the nuclear lamina. These are going to be the fibers that align the inner membrane. So over here, you can see the nuclear lamina. They're the little fibers that line. And then we also have to take a look at the matrix section. The matrix section is just the internal workings. This is what's going to be interacting with the lamina, and it's also going to, in essence, fill the interior part of the nucleus. So what we see happening here is that we have our DNA, we have our nucleus. The DNA has been doing its coiling. It's been making its nucleosome. It's down into the fiber section. You can see right here a close-up of the nuclear matrix. There's all my DNA. How am I going to keep coiling it? Well, it turns out that we have some anchoring that we can utilize. So here in this illustration, it shows you that within the membrane of the nucleus, there are specific sections that can be utilized to excuse me, to link together to the MAR or SAR segments of your DNA. MAR stands for matrix attachment regions. They were formerly known as scaffold attachment regions. These are specific sequences of your DNA that you have within the chromosomes. Well, I shouldn't say chromosomes, my apologies. Within the fiber or the chromatin of your DNA itself. And they're able to recognize and anchor to the nuclear matrix. So here you see this little yellow circles. That is my little anchoring point within my nuclear matrix. And when it anchors, what's going to end up happening is that they'll anchor at a specific location marked by the MAR or the SAR within the DNA itself. And as it does so, it creates these little radial loops. And the radial loops will allow the already compacted DNA to be compressed even further down. Isn't that an interesting concept to go? And what we see happening is that it will keep doing this, and it turns out that each chromosome has a very specific area within the nucleus where its MARs are able to be recognized by the nuclear matrix. So we have these things called chromosome territories, and it turns out that each chromosome has a specific location where its compaction will continue with its MAR sections. So go ahead and take a look at this picture, and it shows you what the chromosomes look like in metaphase when they're all tightly condensed and they're on their way to sit in the equator of the cell. Notice how each chromosome has its own location, and they really don't disperse that much, because if you take a look at the picture on the right-hand side, you see that during interphase, so during day-to-day activity when the cell is just doing like its G1, S, and G2, notice how the chromosomes also have their individual section. Obviously, in this case, they're not as tightly coiled because the genes need to be freely accessible for their day-to-day activity. All right, now as we see the MARs and the SARs interacting with the nuclear lamina and they're coiling up, we see that they're, as the radial loops start to form, you basically get two versions of the compaction. You can have euchromatin and heterochromatin. Well, what's the difference? Well, it turns out, and you can see it on the illustration at the bottom right here, euchromatin is going to be less condensed than heterochromatin. Both of them are utilizing the radial loops. However, the euchromatin tends to not be coiled up as tightly. This is usually correlated with areas that have lots of genes, so they're transcriptionally active, meaning you have easy access to the DNA sequences that need to be utilized. The heterochromatin sections are usually areas that are going to be tightly compact. So these are going to be areas that are not active from a transcriptional standpoint. These are going to be areas that most likely have lots of axons or intergenic regions that we don't really utilize on a day-to-day basis. And as you can see right here in our illustration, they tend to be centered towards the telomere and the centromere section. Now, one thing that I do want to point out is that the when the genome is getting ready to leave interphase and go into mitosis or meiosis and initiate its prophase, metaphase, anaphase, telophase sections, we do see that it becomes essential for the chromosome to be compacted as best as it can. So what this slide primarily points out is that by the time the cell is in metaphase, the level of compaction has to be towards as much as possible as the cell can do. And what we see happening in order to accommodate that is when we were previously talking about the euchromatin versus the heterochromatin, we see that all the sister chromatids will eventually go into the heterochromatin compaction, which is the one that's really super tightly coiled, because as you might recall from your cell cycle, metaphase is when we have to properly align all the sister chromatins down the equator of the cell to make it easier for them to split and go towards their opposite ends in anaphase. So by the time you get to metaphase, all of the DNA is in heterochromatin formation, as tightly coiled as it can to make it easier to manipulate and obviously disperse the DNA as you get ready to go ahead and make your sister chromatins. All right, so here on this illustration, it's going to walk you through how we went from, I'm going to keep calling it naked DNA, because this is just DNA by itself as we're used to seeing it in like DNA models, all the way to the stereotypical chromosomes. So, and this is what our last maybe 10, 12 slides we're chit-chatting about. So on top right here, we have our stereotypical double helix, with our A binded to the T, G bounded to the C, utilizing our hydrogen bonds to do so. And when it's now time to start compacting, our first stop is going to be to form nucleosomes. Nucleosomes is when we take our naked DNA and we wrap it around our octamers. Remember those 8 histones? And there will be a linker region in between with some naked DNA, and that you'll also have your linker histone over there. And as you form your nucleosomes, they'll start stacking on top of each other so that they create more of a 3D structure. So we're thinking chromatin, we're thinking our telephone cord, and we get our 30 nanometer fiber. We're not done yet. That fiber is going to start interacting with the nuclear matrix and the nuclear lamina. It's going to start forming radial loops with the proteins and the MAR and SAR segments of the DNA itself. And as it does so, it can either be coiled up a little loosely, we call that the euchromatin, or more tightly, that's going to be the heterochromatin. Now remember, in a day-to-day scenario, all your genes that are active are going to be in the euchromatin format, whereas areas that are not used, like the centromere and the telomere, are going to be in the heterochromatin format. However, what we do see is that if the entire nucleus needs to be compacted, like in the case of metaphase, all of it will keep coiling until everything is in chromatin, and we get our beautiful stereotypical chromosome appearance that we're so used to seeing when we take a look at things like our karyotype maps. Isn't that beautiful? Let's see. So notice how the top one once again shows you the compaction level at euchromatin versus heterochromatin. Much tightly coiled in heterochromatin. And you know, the interesting fact is that with all this compaction that's going on, if you're ever interested in extracting DNA from a fresh or a live cell and you want to uncoil that DNA, it turns out it's as easy as just sprinkling a little bit of salt on the DNA structure. Salt will go ahead and just remove a lot of the histones and linker proteins, allowing the DNA to quickly uncoil so you can resort back to its naked form. All right, guys, and ladies, that's all I have for this side. Please feel free to email me with any of your comments, questions, or concerns. And as you finish up this part of Unit 2, that means that we are getting ready for our second exam. So please take your time, utilize your Learn Smart modules, take a look at your notations, ask any and all of your questions, because very similar to exam number 1, exam 2 will be heavily based on our PowerPoint and our audio recordings. Alrighty, we'll talk soon.